Hello all, my name is Dr. Bhanupriya. I would like to thank the entire team of this conference for inviting me for this talk, and I would also like to congratulate the other speakers for this. So I precisely work in Sorghum bicolor. Basically, that work is targeted towards the downregulation of the one of the gene in the lignin biosynthetic pathway to reduce the lignin content so that the carbohydrate can be used as a bioethanol, which can be blended with the petrol. So that can be used as a biofuels. So to carry out that work, there should be a defined kind of a plant tissue culture work, which we have tried to optimize. And this is the work what I'm going to present today. So the topic of my work is basically Callus mediated organogenesis and regeneration of sorghum bicolor under the influence of natural and synthetic growth regulators. Okay. So this work was carried out at IIT Kharagpur, West Bengal, India. So sorghum bicolor, it is otherwise called as sweet sorghum. It's a tropical grass species which was initially domesticated as a grain crop in the sub-Saharan African areas. Later, this plant has migrated to India, Assyria, China, and United States also. This plant is a significant crop, energy crop for biofuel production as it stands out because of its rapid growth. It is a marginal crop which doesn't require much kind of resources. It can grow in the drought conditions also, and it is a potential for having a high sugar accumulation because the stalks of this crop is having a good sucrose content, and it has a good amount of biomass which can be used for bioethanol, which is the targeted work of ours, and it has a broader adaptability to the changing environmental conditions. So, to carry out any kind of a plant transformation as we require, a maximum regeneration ability is essential to the successful plant transformation with the target genes. So sorghum is a recalcitrant crop. That is a major problem with this kind of a crop. Uh, the reason is it cannot de-differentiate, which leads to hardening or sclerification of the wounded end, making the transformation process difficult. Apart from that, there is one more problem that this plant is having uh, early rooting tendency also, or production of inhibitory substances, like for example certain kind of secondary metabolites and all those things. So that is one of the common problem what we are seeing with the sorghum. So we have tried from our end to address this kind of issues where an optimized protocol can be developed which can be used for the future uses. So there are two approaches to do that one. The first one is a genotype selection, which is a time consuming process where you have to selectively go for a defined trial and error process to basically narrow down to a certain kind of a genotype from where we can select the explant. This is a time consuming process. The second one is to optimize the plant tissue culture protocol by modification of the media, by certain kind of a growth regulators or nutrients or some kind of supplements and all that. So we have tried the second approach to do this one. So to carry out this work, we have taken three varieties of the sorghum bicolor, that is BSR1, CSV17 and CSH1. So this kind of these three varieties were used and they were grown in the greenhouse, and with a relative humidity of around some 60 to 65 percentage, with regulated kind of humidity levels. And what we have found out is the once the plant has given the mature seeds, we have collected that, we have like sun dried that, and later on the plants were the seeds were surface sterilized with HgCl2 and the tween 20 along with the liquid detergent. And once the seed sterilization has been happened, the next step what we have done is the seed germination. In the right hand side top picture, you can see that the seeds have been germinated. Here, the light colored, the shoot meristem which has been selected and which has been used as the explant. Actually, we have tried our work with two explants. One is the shoot meristem, the other one is the immature embryo. But due to some weird reasons, the immature embryo has given lot of contamination, specifically with the bacteria. That is why it was not an easy process for us to continue with that explant. Rather, we have selected the shoot meristem. So this like shoot meristem, we have continued our work and we have carried out under different kind of the callusing media. So in this, we have designed a set of the callusing media. The basal media, what we have used over here, the topmost one, where we have taken the MS salts, B5 vitamins, along with sucrose, asparagine, and proline with a substratum of the agar. So this was used as a basal media. So here we have used the natural growth regulator. With our trial and errors, we found coconut water is having a good efficiency at around some 5 gram per liter. Along with the natural growth regulator, we have also used some synthetic growth regulators. Here we have used 2,4-D, IAA, and kinetin. So in that, what we have found out is this particular media, that is the basal media, along with 5 grams per liter coconut water and 3 mg per liter of the 2,4-D. This is having an excellent kind of a survival rate of the explant also, and it is having a very good kind of a callus induction rate. So we have continued our work with this kind of this particular media. So once we were doing this the callusing, what we have found out is there are three different sets of the varieties of the callus we have found. The first two pictures, what you're seeing is basically the green friable regenerative callus. Now this one, it is having a very good kind of a regeneration. It is producing the multipolar shootlets and all. The second one, what you're seeing is the rooty callus. Now this particular callus, instead of producing the shoots, it is ending up into production of the roots. So which is not a desirable one for our work. The third variety of the callus, what we have found is basically having a negligible kind of the shootlets. And one more important thing what we have noticed is both the rooty callus and the hard callus is ending up into rotting after some 1 or 2 weeks. Though there is a negligible amount of shootlets also, and it is not growing healthily. I mean to say there is a kind of like contamination also, not by the bacteria, by like producing certain kind of the polyphenols and all those things. So this two calluses we have tried to avoid or ignore. So we have continued our further work with the first type, that is a green friable regenerative callus. And one more important thing is the effect of the callus age on the regeneration. So here, what we have found out is there is a specific time where the callus has to be transferred from the callusing media to the regeneration media. And with our study, what we have found out is all the three varieties have, there is an increase in the callus fresh weight, which was definitely found in all the three varieties, whatever we have used. But what we found out is the callus weekly growth ratio, if we see BSR1, which is marked with the blue color, and CSH1, which is marked with the gray color, these two were growing faster than the CSV line basically. So here in the second week, the callus growth ratio has been increased. And one more thing what we have noticed is that is the regeneration rate also has peaked in the second week only. That means the regeneration efficiency, that means the number of the shoots produced per callus, it was more in the second week, and among the three different kind of the varieties what we used, BSR1 is having the highest efficiency of producing more number of shootlets, which you can see over here in the fourth point. That is, it is having 37 shootlets per a single callus, which is having an efficiency of around some 43%. So we have continued our further work with this variety only. And to make sure that the regeneration happens properly, we have again designed a set of the regeneration media. Here we have used two growth regulators along with the coconut water, that is the benzyl aminopurine and the zeatin. and both at a ratio of 2 gram, that is like 2 mg per liter and 3 mg per liter. And what we have found out is BAP at a 3 mg per liter along with the basal media and the coconut water has shown the highest callus survival rate along with the maximum regeneration rate. So this particular media has been selected. But there was one important issue what we were facing is, which is coming in the next slide, is there is increased secretion of the polyphenols, which is making the entire media dark. So along with the coconut water, we have tried to apply or implement the coconut milk also for this particular work. So this is the amount, what we have selected, and it has been it has played a very significant role in our plant tissue culture protocol. The coconut milk what we have used, it has given us three important kind of benefits. The first one is the polyphenols whatever has been secreted, that has been cleared. So you can see a clear media when we have used the coconut milk and coconut water in our study. The second important thing what we have observed is there is increase in the regeneration. So that increase in the regeneration, that means giving a very good amount of the ratio of the shootlets produced, which has helped us to bringing that regeneration properly, which was done by this. And the third important thing what we have noticed is when we are increasing the coconut milk, it is acting as a compensation or a substitution for the sucrose level. So simultaneously, as the coconut milk has been increased, the sucrose level is also coming down. So once the shootlets have developed, they were provided in a liquid media to produce the roots. So to produce the roots, we have used two different kind of auxins, that is indole acetic acid and naphthalene acetic acid. So when we have used indole, we have found out that the shootlet response were around some 62 percentage to around some 78 percentage. When individually we have used NA, that is naphthalene acetic acid, there is an increase in the percentage of 72 to around some 85 percentage. So both these auxins were used individually. But later on, when we tried to combine the IAA and the NAA, what we have found out is it has given a shootlet response of around some 88 to 100 percentage, which is the maximum percentage. So all the shootlets were able to produce the roots when the both kind of auxins were given. So you can see in this particular picture that there is the roots have been developed. So once the roots have been developed, the shootlets along with the roots were taken and transferred to the soil, right? That is the soil. Now the one important thing what we have done over here is the soil what we have used, it is in combination with the coconut husk. So we had tried to use coconut as far as possible for this particular study. So the third picture what you're seeing over here is the shootlets in the soil, but they are in the controlled conditions. After some 2 weeks, we have tried to after 1 week only, we have tried to adapt the plants in the pot, that is in the field condition, that is in the natural conditions basically. And once they have grown to a decent height, we have transferred them into the ground. That means in the field trials. So this is the outcome of our work. It's a tried and tested plant tissue culture protocol was established with the shoot meristem as the explant. So the issue what we were facing, that is the browning of the media because of the secretion of polyphenols, that has been addressed using the coconut milk. The application of the byproducts of the coconut milk, coconut water, coconut husk has been used really well, and it has allowed us to increase the shoot regeneration efficiency, which is a very much prominent feature when we are going for the plant transformation. And it has used as a compensation to sucrose also. So this is the work. So this is the outcome of the work. And in regard to this, we would like to thank the Nagarjuna Group and the Sri IIT Kharagpur for providing the fund. We would also like to thank our staff of Advanced Lab for Plant Genetic Engineering IIT Kharagpur. Thank you.